Bonjour tout le monde. I'm really happy to be here again. I was here also in 2022 and it was a great event. I'm really looking forward to interaction with all of you and with the other toolboxes as well. As has been explained, we're giving the lectures together. Oh, let me just untangle myself a bit. And each of the presenters is going to cover one aspect. So each of these lectures is going to be toolbox agnostic. At least that's the idea. And then in the practical session, we'll make it toolbox specific. I'm going to explain how to get to know your data, how to start with the whole processing. And I was asked to give a presentation on pre-processing, segmenting, and artifacts. But actually, you could also think of it like in a different order. You could first think about the artifacts, then pre-processing, and then segmenting, or possibly even like first do the segmenting before you do the pre-processing. I think this is a very common challenge that we have in the pre-processing stage, is that you can combine all of these steps that we're doing, combine them in many different orders. And that is sometimes confusing for people that are new in the fields, that there's many orders. And it's not necessary that one is correct and the other one is incorrect. There's reasons why we sometimes do it one way and sometimes another way. And I hope that especially during the practicals that will also become more clear. What I should also say, my slides are online. Actually, all of these lectures will be recorded as well and will be shared afterwards as well. But if you want to look at my slides, here's the QR code, okay? The learning goals that I've set for this lecture is what are the characteristics of the data that we use in the analysis and how to organize your raw data. So I'm going to cover bits as well a little bit. Quality assessment and quality control. And talk about like what are the artifacts and why are artifacts relevant and how should we think about dealing with these artifacts. And then I'll move on into pre-processing and segmenting, or as I already said, like first segmenting and then pre-processing depending on the dataset characteristics, and selective averaging to get event-related potentials or event-related fields. But to start, somehow you have data. You've collected data in your own lab, or you have collected the data from somewhere else, like you have designed your own study, you've recruited your own participants, and you've done the recordings, or you have received data from a former colleague, or you've downloaded it from an online repository. The first thing that you do with your data is you organize it. EEG and MEG data has a lot of different parts to it. It's not just the EEG files, but there's other parts as well. And we often think of EEG as that signal that we record from the brain using our amplifier and it goes onto an acquisition computer. But if you look at an EEG lab, there's way more components to this that are relevant also during the analysis. So we have the participant that is interacting with the stimulus presentation computer. So both ways, the stimulus presentation computer is showing things, the participant is pressing buttons or doing other things to respond to the stimuli. This results in triggers that are being sent to the EEG device. But possibly you might also be like recording events, for example, through LSL that are directly going to the acquisition computer. There might be a stimulus presentation file that is like a log file that is written to disk, which you then copy afterwards from your stimulus presentation computer to your analysis computer. And at the same time, you also have characteristics of your participants, such as the handedness, the age, or other characteristics that are relevant during the analysis. So don't only focus on the EEG file, because there's much more to the EEG experiment than just the recording of the brain activity. So prior to pre-processing, what we do is we do data curation. We collect all the files from all the different computers that were involved. We name them consistently, and there it's important. EEG and MEG data is large. It consists of many files, and it is complex. And that is really something that you have to get used to. It's a challenge that we had also in preparing the practicals. Is like, how do we get these large and complex data sets towards you? Because like even a single pruned subject with some processed files is like between 5 to 15 gigabytes. So you all downloading this was actually quite a challenge for us also to prepare. So if you think of an EEG recording, then a typical recording would have like 64 channels, would be done at 500 hertz, and then 1 hour is approximately 500 megabytes. That easily fits on a USB stick. And then in a typical study would have like 30 subjects or so. That means that we have 15 gigabytes, which still easily fits on a USB stick. And we have many EEG companies, so we have many file formats. And the analysis of these types of data, because they're not that large, they're typically done on laptops, of course, on a desktop computer, but you don't necessarily need very big computers for this. MEG, however, is usually done with many more channels, like 275 or 360, depending on which system you have at 1,000 hertz or so, which means that 1 hour is easily like 4 gigabytes of data. And 1 hour is being quite a typical time for a recording session with a participant. And then again, in a typical study with 30 subjects, this easily amounts to 120 gigabytes of raw data. So then it really becomes more challenging. At least my laptop typically doesn't have 120 gigabytes of disk space free, so I already have to put it on an external disk or so. There's not so many MEG companies, although the number is increasing again. So that means that there's a small number of formats, and that the two systems that are most popular are the Neuromac system, and the CTS system. And with the Neuromac or the Elector or the Megan, so the company has been rebranded a few times. Nowadays they're called Megan, but we often still refer to them as Neuromac. One recording is 1 FIV file, and that's the file format that we'll be working with during the hands-on sessions. With the CTS system, which is also the system that we have at Donnes in Nijmegen, then one recording is one directory, and then in that directory there's a whole bunch of files. The analysis of MEG datasets is often not done on laptops because the data is simply too large, but it's often done on large computers. So we often do this on a compute cluster or on a compute server, and they often have Linux installed. So that's also something for you to get used to, working on bigger computers. One of the really fascinating developments nowadays is optically pumped magnetometers. This is a new type of sensor with which we can record very small magnetic fields, and OPMs, as they're called. They're very small sensors, and you can place them individually on the head. With such an OPM system, so compared to the squid-based MEG system, where all the sensors have to be in a cryogenic dewar, like a helmet-shaped thermos flask, basically, that is filled with liquid helium. The thermos flask or the dewar is fixed. There's only one size, so the participant sticks their head in. If you have small participants, children, the distance from the brain to the sensors is pretty large. With OPMs, you can place them directly on the scalp. Similar like EEG or FNIRS, you place them directly on the scalp. So that means that you can accommodate the different head sizes much more, but also because these sensors are really small, they're lightweight, and you can make MEG recordings while the participant is moving. So this is an example of the, this is an example of, on the left side, it's the Neuromax SWID-based MEG system that we have at the Karolinska Institute in Stockholm. It's a participant that's also wired up with EOG, horizontal, vertical EOG, like ECG is recorded, so that's why there's a whole bunch of wires. There's head localizer coils as well. And on the right, you see in the same lab, you see the OPM system. And this is the OPM system that was initially installed, so it's the trial system. So now the system looks actually quite a bit different. But this OPM system consists of small sensors, and they're really like this large. And each sensor is in a sleeve to make it a little bit easier to slide it in and out. So the sleeves are a little bit longer, but they're basically just like 3D printed sensor holders. And with that, we can slide the sensors towards the scalp surface. So this is one of the systems. So this is the field line system, and we now have a field line system at the Donnes in Nijmegen as well. But you can also get sensors from Serco Magnetics or from QSpin. There's a few companies that are making this system, or Macro Health, which is a French company. And this is an example of the Serco Magnetics OPM system installed in Cardiff. And here you can see how small the sensors are. And what they decided to do is they made like a 3D printed bicycle helmet type of thing. So you wear the helmet and you can move around with it. And which, of course, is really creating unique opportunities for recording MEG while the participant is like in a more naturalistic setting. It is important, though, to mention that OPM is still done inside a magnetically shielded room. So the artifacts that we have with OPMs are the same as the ones that we have with cryogenic, squid-based MEG. So as such, suppressing environmental noise is still important. Why am I saying this? Well, first of all, to get you enthusiastic about OPMs, because I think that's going to be really important new possibilities for our field. But also because OPM data has slightly different characteristics than squid-based MEG data. So an OPM sensor can have 1 or 2 or 3 axes. So either it only measures in one direction, or it can even measure into like 3 different directions. And a total system can have anywhere between 1 to like close to 400 channels. Like you can record MEG with a single channel, and then you can, for example, move it around over the scalp to do like multiple runs. But of course, like it's way more convenient to have a whole head system with many sensors and thereby also many channels. The signal to noise of an OPM system actually pretty comparable to that of a squid-based MEG system. So the size of a study that we would have with an OPM system would still be like probably 30 subjects. So it's still going to be a similar amount of data. But on top of the OPM data, we have to deal with the OPM sensors, which can be placed in a flexible cap. So with a Neuromax system or with the CTF system, the sensors are in a fixed helmet and the position of the sensors is exactly known. With these sensors, you can place them flexibly. And that means that in terms of the data that we collect in the lab, we also have to collect where the sensors are placed. So we have to record the position of the sensors relative to the cap or relative to the helmet. And we have to do different ways of co-registration, for example, using 3D scans of the head and of the sensors, or we have to use a polymers digitizer, etc. So that means that with OPMs, the data handling is still a little bit more complex because there's more files that are being generated to co-register all of this data so that we can do the analysis. So I constantly have this pop up here that about the connection not working. So that's why I often have to click twice to click the thingy away. It's a little bit annoying, so I hope that Adrian can fix this afterwards. So we have all of the data that comes from the primary recording device in the lab. But what I already said is we also have a lot of other computers in the lab that are also generating data. And we can call it auxiliary data. So first of all, we might want to have the participants in the MRI scanner. So we record an anatomical MRI, which when it comes directly from the scanner is represented in DICOM files. So we would typically have 200 DICOM files. But in neuroimaging, almost everyone converts this very quickly to a Nifty format where you would have one file per participant. We have our behavioral data, which is like of course, EEG and MEG are really good in with the temporal precision. So the time-resolved behavioral data is really important. And that is mostly encoded as triggers together with the MEG or EEG data stream. But there might also be stimulus presentation log files on the presentation computer that you need during the analysis of the data. You might also have recorded video or audio recordings of your participants, for example, to see whether they were doing the task correctly or whether they were giving verbal responses. And you might have like an eye tracker. So there's multiple computers involved in a typical lab setup. And then, of course, we often have other data from our participants, such as demographics. And this is often like tabular data. It can be questionnaires, but this data might also be very important in your analysis. So this is not something to forget. So it's not only about the EEG files, it's also about the other files. These types of data, they're typically relatively small. As such, putting them on a USB stick is not a problem, but but they're relatively complex because you can have multiple types of these files. So putting them in the right structure is not always trivial. And this is where, in organizing the data, it is important to organize it fair. Fair stands for findable, accessible, interoperable, and reusable. Findable means to make your data available in a catalog or repository with a persistent identifier and with metadata, so that if you publish on your data, that your reviewers can check that it's really the processing that you did in the way that you described it in the paper, but also that people that read your paper, that they can follow up on your analysis, that they, for example, can use this to inspire their follow-up study. So sharing data is really important, and if you want to share data, your data has to be fair and has to be findable. Of course, your data also has to be accessible, so you have to be explicit about the data usage terms, so the agreement with the downloaders. In some European countries, that is easier than in others, even though we all have the same GDPR. At the donors, we share a lot of our data. I know, like yesterday, we were discussing in Finland, it's still extremely difficult to share data, but it is important that you agree with your participants and that you also agree with your ethics department or your ethics team about like how much you can share the data. The more open, the more science will benefit. Then there's the more technical aspect of interoperability. So interoperable means that someone else can also read your data. So you have to make your data human and machine readable. And you want the data to be reusable, so that means that you want to make sure that you document enough details. This can, for example, be with the data descriptive paper, as we have for the for the data set that we're using throughout this week. And the data descriptive paper is really cool because it can be cited. So as such, it creates like measurable impact. It's going to be relevant for your CV and for your career. BITS is one of these very important steps that we've been working on for the last 10 years. And it's a community initiative to make your data more fair. BITS is a way to organize your existing raw data and to improve consistency and to complete the documentation, and thereby to facilitate reuse by yourself and by others. And reuse by yourself is really important because while you're recording data, there's a lot of details. And perhaps you're only going to be looking at those details half a year or a year after you've collected the data. Or sometimes there's even more time between you being in the lab and you doing the analysis. So keeping track of all the details in the lab, that's where BITS helps. And BITS is not a new file format. It's not a search engine or it's not a data sharing platform. So those are other things. But BITS is really about the increasing the interoperability and the reusability of your data set. So this week we're working with a BITS data set that has been prepared for you. But what's important is that you also start considering like how do you make your data BITS compliant and how do you share your data? So perhaps the next time that we're going to do a practical MEG event, we're going to be analyzing one of your data sets rather than the one that was recorded in 2015. As Anso already said, BITS in principle is just a bunch of files and directories on disk. There's not really special software needed, although there are tools available to work more easily with BITS datasets. And this is an example of a BITS dataset. And what you can recognize is that there's a specific directory structure that is standardized. There's the actual EEG data, and this is here it's an EDF file, but this comes directly from the amplifier. But there's also a whole bunch of files around it which have the metadata that have information about the participants, the EEG recording device, events, etc. And in BITS terminology, we call these sidecar files. They're basically files that stand next to the main files. And they represent otherwise missing data, and they make it easier to query or to search. So just to give an example for EEG and for MEGs, we have a TSV file, so tab separated values file, which you can just read in Excel with details about the participants. We can have the same thing for the sessions, for the scans. And we, for example, have a file in which the details of the MEG recording machine are specified. With MEG, the files are pretty complete, so there's not too much that we're actually missing, but especially with EEG, and if you use an EEG system that spits out an EDF file, based on the EDF file, you cannot even see with which system it was recorded, or where the electrodes were placed, or what the reference was, or those type of things. And those are the things that we have in the MEG.json or the EEG.json file. Then also very important, we have an events.tsv file. As you know, the EEG file or the MEG file has trigger codes, and these trigger codes are numeric. And it's in your stimulus presentation that you translate the stimuli that you're presenting, so the different types of faces, the unfamiliar or the famous or the scrambled faces, you translate it to different trigger codes. And here you can see the column with all the triggers, but you can also see what type of face and whether it was the first or the second presentation. All of these things can be figured out on the basis of an events.tsv file. So an events.tsv file makes your data much easier interpretable because there's a lot of details in your experimental task that otherwise are only represented in numbers, and figuring out those numbers is difficult, whereas an events.tsv file is something you double click and then you see it and you think, ah, that makes sense. Having dealt with what the characteristics of the data that we're working with EEG or with MEG and how to organize, and I want to move on with quality assessment and quality control, and what are artifacts and why are they relevant and how do we deal with them. Pre-processing is one of these things in analysis where actually it's often not so clear where the pre-processing ends and where the processing starts, or where the analysis starts. What I often try to think about is like the pre-processing is that part of the analysis, like that part of the work that you do on your data, which does not depend too much on your research question. So quality assessment, looking at artifacts, segmenting, aligning the MRI with the EEG or MEG sensors, all of those things you would do in any type of recording. But the later steps are more tightly linked to the research question that you have. So whether you're going to average event-related potentials, or whether you're going to compute power spectra, whether you're going to do source modeling, all of those are more like research question specific, are more optional. And I would say that's more the processing or the analysis side, whereas the pre-processing is the stuff that everyone does. And what I want to do is I'll highlight a few of the steps from the pre-processing and one of the steps of the analysis to just show how we go through this. It is important to plan your analysis. And very often what happens is that we take 2 steps forward and 1 step back. So if you're gonna, so for me, this is one of the reasons that I like scripting. I like to write my analysis in a script because then I can easily go back and I can re-execute part of the script, make some changes, and then re-execute it once more. So this is something that you should plan for. How are you going to organize your data? How are you going to keep your scripts separate from your data? How are you going to keep your process data separate from your raw data? So I typically have 3 directories: raw data, process data, and scripts. And in my scripts directory, I usually have different scripts, like different versions of the scripts and different scripts for the different steps. So plan for multiple iterations. And it is very common that it's going to look like this. Like, it's not necessary that you're going to take a straight path towards the end of your analysis. You really might have to backtrack. You might have to backtrack multiple times. And then in like you evaluating the results that you're getting, you realize, oh, I really have to go further back and redo part of the analysis to move forward. It helps if you have a clear goal. So if you know where you're starting from, if you know where you want to end up, and then you have to find your way from the starting point to the end. So that helps in terms of thinking about how you organize your scripts. And it's still like everyone's going to write the scripts in a different way. Everyone's going to organize their analysis in a different way. But this is basically what we're all doing. One of the very first steps in working with your data is quality control and to deal with artifacts. And some artifacts are really simple. Like with EEG, we have electrodes that are attached to the subject's head. If we have a bad attachment, we have a bad signal. With MEG, we don't directly attach the sensors to the head, so the OPMs are like on the scalp, but they don't touch per se. Like there can be a little bit of padding in between. And it means that with MEG, the quality of the signal does not depend on how well the sensor touches the scalp. And the number of bad channels that we have, that often depends only on the hardware, not necessarily on how you've prepared the subject. Sometimes with our CTF system in Nijmegen, at this moment, I think we have 4 broken channels. Well, they're broken for a few months, and in November, we're going to do a maintenance, and hopefully during the maintenance, we can fix them. But it basically means that for like 6 months or so, we have the same 4 broken channels, and it doesn't depend on the participant. But of course, like the more tricky ones are the ones where we have artifacts. And in EEG, artifacts are anything that causes potential differences, like electrical voltage differences. And with MEG, artifacts are anything that causes magnetic fields, except those magnetic fields that we record from the brain. And it is important to be aware that the size of the EEG signals is very small compared to a battery. And that order of magnitude is the same as a battery compared to lightning. So it is as if we're trying to measure a battery while lightning strikes, or we're trying to measure EEG while the participant is holding a battery. And these 1 voltage type of things, or 1.5 voltage, actually they're very easy to generate by just rubbing your feet over the carpet, etc. So there's a few artifacts that we have with EEG. Like one of them is poor contact with the scalp. But then we also have like electrochemical noise. So if the participant starts sweating, the ion concentration between the scalp and the electrodes changes, and that works in the same way as a potato clock. Like if you just stick 2 different pieces of metal in a potato or in an orange or an apple, whatever, you can measure voltage difference, and the voltage difference is about, like with nickel and copper, it's about 0.7 volts. So with 2 of those, you basically have a battery that can run a clock. We have the same thing on the scalp. So we also have a potential difference between the scalp and the electrode, and that can be challenging. Electrostatic noise, the participant rubbing their feet over the carpet, like especially in winter when it's really dry, it happens, then your hair can stand up. That's not a volt. That's easily 10 or 100 volts. Like if you touch something, you can actually feel the discharge. You can hear the ticking sounds. So those are artifacts that we have while we're recording EEG, and those artifacts are way larger than the microvolts that we're trying to record. We also have artifacts that come from the environment, such as power line noise, like from all the electrical sockets around it, the 50 hertz equipment, computer screens, etc. And then we have other types of physiological activity that also generate bioelectricity. So we have muscle activity. We have the heart, which of course is also a big muscle, but like which contracts very regularly. We have eye movements, and all of those contribute to artifacts that we need to think about. One artifact that's often not so well understood is electrode movement. If the participant makes a jerky movement, then it might happen that due to the cable pulling on the electrodes, that the electrodes move a little bit relative to the scalp. And what we have is we have the skin, we have the head, and inside the head, electricity is represented on ions. So we have chloride, we have potassium, we have sodium, we have calcium, and it's those ions that allow us to move around. Of course, those ions are also over the cell membrane, and that's how neurons work. But in the electrode, we have electrons. We don't have ions. It's basically a metal wire, and it's on the interface between the skin and the electrodes. That's where we have an electrolyte. So we need something in between that translates these ionic currents into electric or galvanic currents. So that means that in this layer of electrolyte, this layer of electrogel or paste or water or sweat, like if you're using dry electrodes, you're just relying on the sweat providing this layer. We have something with ions. If the electrode moves, you're messing around with the ions. So all of a sudden, this small layer of ions is totally disturbed, and that causes a large artifact. So this is why, like the thinner the layer is with dry electrodes, the more problematic it is when you start having movements with the EEG electrodes relative to the scalp. With MEG, we have a similar order of magnitude thing. So on the one hand, we have the MRI scanners, which are about like order of magnitude of Teslas. Then we have an Earth magnetic field, which we consider like a very normal field. Like it's also here at this moment. It's 10 to the minus 5 Tesla. Then we have the human brain, which is actually like more than 1 million times weaker than the Earth magnetic fields. Like it's in the order of pico or even femto Teslas. And in order to record MEG, without being disturbed by like large artifacts such as MRI magnets, fridge magnets or so, or the Earth magnetic fields, we have to do these recordings in a magnetically shielded room. So this is like the old magnetically shielded room built at the MIT by David Cohen. And nowadays they're like constructed differently. They look like rooms, but you might have seen them. And nowadays it's actually getting more common again that we combine this with active shielding by putting a Helmholtz coil around the room or by putting them inside the rooms. It's something that you might have seen also from the OPM photos that I showed before, is that we have these planar nulling coils. Common MEG artifacts are those that come from outside the room. So we have like power line noise, like the electrical equipment outside of the room. That's often 50 hertz, but also large metallic objects moving outside the MSR. Like cars driving on the streets, elevators are often a challenge. Like if you have a big chunk of metal moving up and down, that's something that we can see in the MEG system. The fan of an air conditioning can be a challenge. We also have the Earth magnetic field, and the Earth magnetic field itself is extremely constant. Like it very slowly changes over decades, but in the course of a measurement, it's really constant. But the problem is that we have this MSR, and if the building vibrates a little bit, or if the walls of the MSR vibrate a little bit, then the shielding of the MSR vibrates as well. So that means that the MSR has to be really sturdy. Most of them are. But still, you're going to see like small vibrations of the MSR that translate into small fluctuations in the residual fields that is due to the Earth. That's one of the artifacts that we can see. And again, as we have with EEG, we have the participant themselves. So the participant has muscles. Muscles work with electricity. So that means that the muscles produce magnetic fields. The heart produces magnetic fields. The eye movements produce magnetic fields. One of the new challenges that we have to deal with is that with OPMs, we can do MEG recordings while the participant is moving. Conventional squid systems, squid-based MEG systems, they have a fixed helmet in the middle of the room. It doesn't move. With an OPM, if a participant is moving their head and then the sensors are moving along with the participant, you become sensitive for differences in the magnetic field over space. So we have residual gradients. Magnetic field is slightly different from one to another location. So if I move my head, you're going to see that difference in the magnetic field. And also if the participant makes a turn with the head, then the direction of the field that we're recording can change 90 degrees. So all of a sudden we're measuring the magnetic field, we're not measuring it. So these head movement related artifacts with OPMs, they can be very large. Luckily, we have very good methods to deal with them, but this is like a very important source of artifacts with magnetometers, and especially with magnetometers that can move. So what we typically do with EEG and MEG artifacts is we try to remove them. So we identify and remove bad channels, or sometimes with EEG we have good reasons to not remove the channel, but to interpolate it on the basis of the neighbors. We also identify and remove bad segments. So if we can look at the continuous data, we can identify parts of the continuous data, where, for example, the participant was moving a lot, or where the participant was like blinking a lot. Or we can first look at the segments, and then for each of the segments, we can decide whether the participant was blinking around the time of the stimulus presentation, for example. And this is what the order of processing is a little bit different. With EEG, it's quite easy to read all the data in memory to work with continuous data. With MEG, the data is often large. So what we often do with MEG is that we only read the segments that we're really interested in, and that we don't necessarily look in between the trials. So that really has to do with the size of the RAM of the computer and being able to quickly browse through so many channels. But it's important to recognize that we're identifying trials in which the behavior was incorrect by the participant doing something that the participant was not told to do, such as moving. Like we often tell the participant to sit still. So moving is actually deviating from that, and moving creates artifacts, or in which the data cannot be recovered. And it's the behavior that is really important here, because these artifacts are not only a nuisance on the signal, but they tell us something about what's happening in the lab. And you can think of the brain as a hierarchical functional network, and there's both sequential and parallel processing. We have both feed-forward and feedback processing. So if we're presenting a face on a computer screen in front of the participants, there's going to be like a whole sequence of activations is going to happen. So we're going to have like edges and lines being detected in V1, and then shapes in V4, and then faces and objects are recognized. And then the decision would be made, like whether the face is familiar or not. And then, for example, we could have a motor response in return. And it's this whole sequence of processing that we can use, because our experimental task and our behavioral readouts, they ensure that we're tapping into the desired cognitive processes. And if you think of, like, for example, a baby EEG study, if the infant is looking away from the screen, they're not seeing the stimulus. So even though the infant might be sitting still looking away from the screen, that's a part of the data that you cannot use during your analysis. So that's why we often record video along with baby EEG, so that we can score whether the infant was actually doing the task or not. If you have an adult participant, if you tell them to look at the screen, well, they typically will do. But if they blink exactly at the moment that the stimulus appears, well, probably they didn't see the stimulus properly. So the blink at that moment is not only a nuisance in the signal, but it actually means that the subsequent processing in the cortical hierarchy that you're studying, in the cognitive hierarchy that you're studying, I should say, is very different. And if the subject doesn't show a response in the stimulus response task, it means that the stimulus was processed differently. Or if the participant responds too slow, then probably there was a different cognitive process interfering. So it's the behavior of the participant, both in terms of the button presses, but also in terms of the artifact that the participant generates, allows us to infer whether the desired cognitive processes were actually active. So the experimental tasks that we design, they often involve attention monitoring. They include catch trials. They include a task that is necessary to make sure that the participant is paying attention. Or they have like extra condition with responses. And these behavioral responses, or the artifacts that you can see as a sign of behavior, they need to be analyzed. So it's not only about cleaning the data, it's about cleaning the task. And then only after having rejected the data that corresponds to bad behavior, or the data that corresponds to broken data, like where really the sensor is not attached to the scalp, etc., we proceed to cleaning the remainder. And cleaning then is really about taking the data and trying to subtract the artifacts, trying to make the impact of the artifacts smaller. Here we basically use the biophysics. So the data is a spatial temporal mixing of different sources that contribute to the data. And we use spatial temporal models to separate the brain and the noise source. And we have different models for EEG and for MEG. So for EEG, independent component analysis or principal component analysis, can be used. And we have like more sophisticated methods such as IC label or automatic ASR, MARA, JEDI. So those are all methods that use the spatial temporal patterns in the data, and either they can do like this like more manually, where you basically have to judge which components are artifactual and which ones are not, or they do it more automatically towards the right side of the table. And for MEG, we have similar but slightly different methods. So with the CTS system, we can use third-order synthetic gradients. With the NeurMax system, we would often use SSS or TSSS, which is also called max filter. It's like that's the name of the software that we use for that. We have signal space projection, and now for OPMs, where we have new types of noise because of the head movements. We also have new types of denoising strategies, such as homogeneous field correction or adaptive multiple modeling. And also for MEG, we can use ICA, but since the head can move relative to the sensors in old-fashioned systems, or since the head can move relative, or the sensors can move relative to space, I would be less confident about like ICA being a robust technique for MEG. But ICA is very robust for EEG, but for MEG it might or might not work. So that's why it's only towards the end. And these cleaning techniques, they're based on data-driven. So like ICA, like blind source separation, it makes assumptions on the data, or they're based on biophysical models, which is like more the case with max filter and with homogeneous field correction. And they make these models on the spatial distribution and the temporal distribution of the brain activity and slash or of the noise. Okay, so I'm not going to explain in detail how any of these cleaning methods work. That's a little bit more for the practicals. But we're now basically at the level that we have done our quality assessments. We have looked at our data, we've decided which parts to keep, and we've cleaned the rest. And we've thought about the relevance of the artifacts. So now we can move on more with the processing or with the analysis. And the pre-processing, that's you could say that's the first step, but then we move on with segmenting. And as I already said, sometimes we only process the data segments that we've read or vice versa. And what I want to go to is like, how do we do selective averaging to get event-related potentials or event-related fields? So I'm working at a Center for Cognitive Neuroimaging. So we always study the brain activity in a task that the participant is doing. And that task is often like an event-related task. There's a stimulus, we know when the stimulus happens, and it's around the moment of the stimulus that we analyze brain activity. Of course, that's not always the case. You might also have like a task-free environment, like resting state, or you might have that the participant is doing a task which is more continuous, such as watching a video for a certain amount of time and then watching another video for another certain amount of time, or where the task would be like open your eyes and close your eyes. But in an event-related task, you know something about the timing. And it's important to realize that the signal to noise of the recordings that we can get either with EEG or with MEG, that's not sufficient to directly observe the brain response. If you have intracranial recordings, as we sometimes have with epilepsy patients, with an electrode that's directly in the cortex, you can see on a single trial, you can see quite a lot. And of course, with brain-computer interface, we're trying to go towards systems that can actually analyze the data on single trials. But in general, the signal to noise ratio is not sufficient. And that's why we already design our tasks such that the stimulus or the task is repeated many, many times or many trials. So for example, we can have one trial every 400 seconds. And then if we have 1 hour of recording, which is typically the attention span of young adult participants, we would get 900 trials in 1 hour. The experimental manipulation is usually only a subtle difference between the trials. The EEG or MEG response of interest is only like 1 second around the stimulus. So that means that 1 hour of recording only results in about 900 seconds of useful data. Like it's only 1/5 of the data or 1/4 of the data that we're keeping for the analysis. This is the data set that we're going to work with this week. So that consists of stimuli that are presented, it's faces or scrambled faces, and some of the faces are familiar, some of the faces are unfamiliar. And you can think of this as, we have 900 trials, and it's 900 times a picture of something. And if you're interested in visual processing, like visual perception, then just presenting visual stimuli might be enough. But here, actually, there's more to the question, because the 900 trials can be split in 600 faces, 300 of which are unfamiliar, and 300 of which are familiar, and 300 scrambled faces. And there's even more to it. So we can have 900 trials where 150 are unfamiliar faces that were presented for the first time, and then the same 150 faces are presented again, but then you've just seen them. like 15 minutes ago or 10 minutes ago, you've seen the same face. So it's not totally unfamiliar anymore. Whereas with the familiar faces, if you see them the first time and then you see them the second time, well, you've seen them so many times, probably the cognitive processing will be different. And of course, there's also the scrambled faces that are being presented. So you can think of this you can basically put those trials in a matrix where we have unfamiliar, familiar, scrambled, where we have the first presentation and the second presentation. But there's actually also many other dimensions in the stimulus material. Like, what's the gender of the face that is presented? What's the emotional expression? Like, is it a happy face? Is it a neutral face? What is the gaze direction of the face? Like, is the face looking straight at you? Is the face looking away? All of those are things that you could study. And this allows for a multifactorial design where you would have stimulus times presentation times gender times emotional expression times gaze direction plus times many things. And then we can start thinking about, like, how do we analyze this? And of course, if we have a multifactorial design, then this is the logical way of thinking about it, like thinking about ANOVAs. So we can look at the main effect of faces versus scrambled faces by pooling all the faces together, regardless of whether they're familiar or unfamiliar, regardless of whether they're presented the first or the second time, and we contrast that with the scrambled faces. Or we basically ignore the scrambled faces and we contrast the familiar and the unfamiliar faces with each other. Or we look at familiarity effects, like, have you seen the face before? Have you seen the face recently before? So first versus second presentation. And then usually it becomes interesting at the level that we start doing interactions. Because we really have expectations that an unfamiliar face is something that you can, like, store in memory, whereas the familiar face is recalled from memory. So we can look at familiarity effects. So this is something that's represented in our data. And here I'm showing a piece of the data. What you can see is that, so this is all the EEG channels. So it's about 64 channels. You see the time courses, and you can directly observe that there are certain like regions where you would have, or you don't see my mouse, but you can see them. You have eye blinks. Also towards the end of the piece of data that I'm showing, there is something which I suspect to be muscle activity. It's like it's shared over all electrodes. It's high frequency. So probably the participant is like moving their mouth or so. Something that is like slightly less visible is that we have these triggers. And these triggers indicate exactly when the participant was doing something. And it's, so these triggers, they basically come from the stimulus presentation computer. They're recorded by the EEG system. And using these triggers, we can define segments where we take a little bit of time before the stimulus was presented, a little bit more time after the stimulus was presented. And each of these is a trial. All right. It also becomes a little bit more clear why sometimes we do the pre-processing on the continuous data, and sometimes we do the pre-processing on the segmented data. Because actually, if we tell the participants that it's not a problem to blink in between trials, then also I don't worry about the trials, about the eye blinks in between trials. Like, that's the participant actually doing what he's supposed to do. So I don't have to clean those parts of the data necessarily. Or if the participant is making movements during a pause, like when there's no stimuli being presented, well, that's not something that I worry about. So that's why for large data sets, the strategy is often that we first segment and then start looking for artifacts. Whereas for smaller data sets where you can, like, more easily get an overview of the whole data set, that's just looking at the whole data, and then you recognize the pauses, you can recognize the moments when the participant is blinking, and you might also see that the participant is actually blinking exactly when the stimulus was presented, and of course those are the ones that we have to deal with. So each of these blocks of data, and I'm just going to squeeze them a little bit smaller so that I can fit more on my screen. So we have about 900 trials. Each trial has about like 64 channels for the EEG, and it's 1 second, so sample at 1100 samples per second. So that means that each of these recordings is 900 times 64 times 1100 is about 63 million numbers. And that's what we basically have to work with. And on top of that, we have the subject, so we have this times 16. So that's why EEG and MEG data analysis, we really have to work with a lot of data. And that's where we start using the structure that we have in the data. So if I now like zoom in on the trials a little bit, so we have channels by time. One of the things that we can do is we can look at a specific channel, which is one specific row of this matrix. And each channel basically has, relative to its time, has the voltage measured. And that's what I'm showing here. So here you see for subsequent trials, you're seeing the time course of one EEG channel, and the logical thing to do is to start averaging them. So if we take the first trial and only copy that, we don't see much. But if we start summing the trials and dividing them by the number of trials that we have, the more we put into the average, the more clear it's going to become that there's something that is shared between all of the raw data, and there's also something that is not shared, and that's what we call the noise. And noise is not necessarily like noise in the sense of like environmental noise. It can also be brain noise. It can also be the participant thinking about things that are unrelated to the task, like what to prepare for dinner tonight or whatever. But event-related potentials allow us to zoom in on those parts of the data that are consistent and that are equal over trials. So that's the assumption, that there's something that is the same in all the trials, and that the noise, the thing that is different in all the trials, it's going to get smaller and smaller when we average. So the brain signal of interest is assumed to be constant over trials, the noise is independent over trials. Now, after averaging, we can make a prediction how much smaller the noise is going to be. And that's proportional to 1 divided by the square root of the number of trials. So that means that if I take 10 trials, my noise decreases with square root 10, which is about a factor of 3. And if we take 100 trials, my noise decreases with a factor of 10. It's very frustrating because it's very inefficient. Like 100 trials and the noise only decreases with a factor of 10. But that's just what we have to deal with. So that's why our recordings are relatively long. What is important, though, is to realize that it's not a signal that is getting larger, but it's the noise that is getting smaller. Again, so this is something we're presenting stimuli. We are looking at these event-related potentials, but we know that there's many different brain regions involved in this. And if we then consider how these brain regions, how they translate into the signals that we're recording, well, then we could say we, for example, have like one region in occipital cortex, the red region, and we have one region in more frontal, and they would have different time courses. So, for example, this having like a low frequency and the other one having high frequencies. And then in our recording, a channel that's more or less in between would see a little bit of both sources. So we see a mixture of the low frequency and the high frequency, whereas a channel that's more on the back would see more of the low frequency, and a channel at the front would see more of the high frequency. So the data that we work with, the channel level data that we work with, is this spatial temporal mixture of the underlying sources. And our research questions are about the brain, are not about the data. So what we want to do, we want to make an inference about what the brain is doing. So averaging the noise over many trials makes the noise smaller, and it keeps the signal the same. So the data, we think of it as the data being signal plus noise, and then we can think of the data in 2 conditions. So we have the signal in condition 1, we have the signal in condition 2, and both of them have noise. And the signal is basically, it's like something that we repeat over many trials. So the signal is related to the task, it's constant over trials, the noise is not related to the task. So by averaging, the signal stays the same, the noise gets smaller. That's the idea. Something that is then very important is we can compute these event-related potentials, and we can start looking at the differences. So we can, by looking at the difference between 2 event-related potentials, we're looking at the difference between the 2 signals. And this is where linear superposition is really important, so that we have this linear addition of all the different components that we have in the data. Because you could say, well, we have the visual processing and visual cortex. And the visual processing for one face is the same, for a familiar face is the same as for an unfamiliar face. You could even say the visual processing for a stimulus, like an oval stimulus appearing on the screen, is the same as it is for a scrambled face as it is for a real face. So we have the visual processing, which doesn't really differ between the conditions, but then we have the recognition. Like, is it a familiar face, is it an unfamiliar face? That is different. And by us looking at ERP difference waves, we can basically cancel out the effects that are common over, that are identical over conditions, and we can start zooming in on those parts of the data that we really care about, which is the face processing and the face recognition. So that means that by looking into ERP difference waves, we can tap into specific cognitive processes. So that's how our analysis links back to our experimental design. And that allows us, for example, to study a faces versus scrambled faces effect, like the N170 that Anso already described, or the familiarity effect, which is like more visible slightly later in the time. And of course, this is where this would then be the starting point also for further analysis in which we can explore this more. For example, asking questions like where in the brain is the face processing happening versus where in the brain is the recognition of the familiarity happening. So this is not the end point, but this is basically like one of the first steps that you do in your analysis. So what I've now done is I've basically talked you over the characteristics of the data that we have in the analysis, that it's important to not only think about the EEG recording, but also about all of these other smaller files that we're recording in the lab while the participants are doing things, such as with the eye tracker, with the behavior, with the video cameras, and that we organize this data. And it's really important that you also start organizing your data in bits. Like bits is really used internationally a lot. It's not necessarily easy to get into, but once you've done it, if you've done it the first time, actually the second time is pretty trivial. Bits is not complex. You just have to get used to it. And if you do bits, then your lab mates and your future self are going to benefit a lot, because your lab mates will have it much easier to start reusing your data. And I'm pretty sure that you will all recall that when you started your experiments, probably you wished that there was a data set that you could try out things on. And hopefully you would get that data set from your lab mate. So by you converting your data to bits, you're preparing the next generation of neuroscientific researchers, and you're helping them to continue on the research line that you have initiated. I've talked about quality assessment. Quality assessment is really like looking at the data, and quality control, where quality control is more about deciding what you do with it, deciding whether you reject pieces of data because the behavior of the participant was not good, or where you decide that the data can be cleaned, that it doesn't interfere, that it's not a confound, but that it's just like a nuisance that you can remove or subtract using ICA, using filtering, or those type of techniques. I've explained a little bit that artifacts are more than just a nuisance, that artifacts tell you something about the behavior of the participant, and that the behavior of the participant is important, especially in cognitive tasks. And very shortly, I've talked about pre-processing and segmenting, or vice versa. I hope that I've also touched enough on why do we do the processing sometimes in a different order, and it's not necessarily that one is correct, the other one is incorrect. It's just more from a convenience perspective that we sometimes swap the order of the steps in the pre-processing pipelines. And then towards the end, I've showed you how we use selective averaging, like selecting over conditions, and how we can use difference waves or ERP difference waves to cancel out effects that are common across conditions, and to look at the effects that are different across conditions, how we really can use our EEG to understand those parts of the experimental task that we're interested in. And with that, I would like to thank you for your attention. I'm looking forward to your questions.